Hey guys, today we're going to talk about enzymes, specifically the higher level content. Now, enzymes is something you've probably all encountered before, lower down in the school when you've been studying biology. There's some awesome demonstrations like elephant's toothpaste that have been amplified on social media where they add a small amount of a liquid to like a bathtub and you see this huge explosion of foam. Now, that's because enzymes, they're lowering the activation energy required for a reaction to take place. So if we look here in this graph, now here is the energy required, and we're going to lower that amount by having that enzyme present. It's what we call a biological catalyst, and it lowers the bar to entry. Basically, it means that the amount of energy required to break a bond or form a bond is therefore less. So, if you remember when we spoke about metabolism in general or biological molecules, we've used the words anabolism, so building things up, and ana the builder, an anabolic reaction, and catabolism, so breakdancing cats is the way I remember that, and catabolism is breaking it down, and that means the addition of water, so like a hydrolysis reaction. Whereas an anabolism would be a when we're building something up a condensation reaction. So that elephant's toothpaste one, the foam being produced, what that is something called hydrogen peroxide, and that's being broken down into oxygen and water. So that is a decomposition reaction. We've got a hydrolysis, water's being added, and it is catabolic. So up at the top of your interactive lesson that you can follow along with this, we've got the three basic concepts, like what they are, how they work, and where they act. And what we're going to focus on, we spoke about these two already. They are biological catalysts. They work by lowering activation energy. And where is all this activity happening? So it's to do with the active site. There's a couple of models. There's an induced fit, where that means that the interaction with a substrate causes a change in the shape, and therefore the active site changes shape to fit it. Or there's the lock and key, where every active site is the exact fit for that particular substrate. So we say that enzymes, for this reason, are specific. Like most enzymes will only work with one set substrate. So, amylase, protease. Amylase is working with amylose. Protease is working with proteins. You'll notice they both end in ASE. So that's one way to tell if what you're reading is an enzyme, because it has ASE at the end. And if we watch this little animation here, see our substrate comes along, the active site changes shape, a bond is broken, and we have some products form. And you've probably covered that before lower down in the school. That's the basic concept of an enzyme. And we always think about them breaking stuff down, so a catabolic reaction. But they also build stuff up too. And that would be dehydration or condensation reaction, removing water from a little bit from each of the molecules that are being added together. So there's a few other things to think about when we think about the basics of enzymes. And the shape is everything. That means they are specific. And if there is a huge change to their surroundings, then that can actually cause a conformational change, which leads to denaturation. When I say conformational change, like the way that it's arranged. So extreme pHs, which means either higher or lower, can alter the shape of an enzyme. It interacts with those R group interactions between adjacent amino acids. And if you don't know what I mean by an R group interaction, you want to watch the video on proteins and protein folding. So the R group will determine the three-dimensional shape. And you have hydrophobic and hydrophilic regions. And you'll probably know that a high pH is basic. That means there's more OH minus molecules knocking around, hydroxide ions, and they've got a negative charge, and they're going to end up going in and causing some disruption, and that is irreversible. And with hydrogen ions, so a H plus ion, a positive hydrogen ion, they are becoming more and more present as we go to a low pH, like pH 1 has a lot more. It's like a logarithmic scale. You don't, as a biologist, you don't need to really get into that detail, but it's this idea that we've got hydrogen ions that are going to disrupt the R group interactions and cause conformational changes. So temperature works in a similar way of denaturation, but that's an extreme. Down here, we've got low kinetic activity. So that means the movement of the particles. They're not going to collide successfully as much. We need that collision, that enzyme coming together with a substrate of a certain amount of energy in order for reaction to occur. There is an optimum amount in our body. Of course, it's around 35.5 degrees centigrade because that's what our homeostasis level is. But if you start going to extreme, then it's going to cause denaturation of proteins, denaturation of enzymes, and then the reactions will stop occurring. And that's why, like, a fever is really dangerous for a small baby. It's not so dangerous when you're an adult, but it can still be fatal because it's going to start disrupting metabolic pathways like respiration, which are dependent upon multiple enzymes. And they have an optimum. And past that point, they're going to denature. You won't get enough ATP, and it can lead to death. And substrate, what happens here is as we increase the amount of substrate, we're going to increase the rate of reaction. But at a certain point, every enzyme is occupied. So if you think there's like 10 people available to catch a ball and I throw 10 balls, well, if I threw an 11th ball, there was no one to catch it. So even though I increase the amount of substrate, that 11th ball can't get caught or that additional substrate can't get used. So the way to think about it is we say that it is saturated. That means, another way to think about the word saturated would be with like a sponge. When it is like holding as much water as possible, we would say it's saturated. And this means all the enzymes here are busy. They are working and breaking down a substrate. And if I introduce more, it's not going to have an effect. Let's move on and think about the HL stuff. This is where it gets interesting. So enzymatic pathways could be linear, A to B to C to D. So each part needs a new enzyme. And what's interesting here is when we can have end product inhibition. So we don't want to keep making something forever within our cells. There's going to be an optimum amount of it. And that's where this guy here, D, can become the inhibitor for the first enzyme. So the arrow should point here. It's inhibiting this. And we can also, so we have linear pathways, but we also have cyclic pathways like the Krebs cycle and the Calvin cycle. And this is a way of managing to keep produce something and recycling some substrates too, which you'll learn more about the respiration as the course goes on. Now, when we say inhibition, that's an interesting word. Like, what does that mean? Inhibit means to stop. And there's two ways to switch off an enzyme. You can have competitive inhibition. So this is my active site. And the competitive inhibitor is the same shape as the substrate and will bind to that, rendering it unable to react with the substrate. And you can have non-competitive inhibition, binding somewhere other than the active site. We call that area the allosteric site. Now, a statin, you might have heard of this before. Statin is to help lower cholesterol levels, and it's a very widely prescribed drug. When people get over the age of 40 and they start having high cholesterol, statins can be introduced to help lower the risk of heart disease, and it works as a competitive inhibitor. That means some proteins, some enzymes are being turned off. And then we have allosteric inhibition or non-competitive inhibition. And an example here, like isoleucine inhibits the first enzyme in its own pathway in its production there. So this is where that addition, if you remember the idea, if we add something or take something away, like I add an extra amino acid, it's going to change the growth structure of the protein. Or like sickle cell anemia, we made that one amino acid change and we went from a normal hemoglobin shape to the sickle cell. And that's going to have, therefore, a knock-on effect and cause all the R groups to interact differently, and the active site itself changes shape. So it will no longer work. Now, we can graph this. We have no inhibitor. This is our maximum amount as we increase substrate concentration. So it will keep going up if there are available enzymes. Now, if I remove some of those enzymes, like completely non-competitive inhibition, that lowers my maximum rate, because if I have, like 10 people available to catch a ball and I've tied 5 of their hands behind their back, well, only 5 now are able to catch the ball. Whereas now I've lowered the maximum amount of balls that could be caught. Whereas you could think of like the competitive one would be I'm throwing like 2 objects to someone and this idea that I can outcompete a competitive inhibitor by increasing substrate concentration. The analogy I use in my seniors, and if this line extended, it would go all the way back up to Vmax. But the analogy I use in my seniors is thinking like about vodka, like how it has a big effect in a when there is a small amount of water. If water is my substrate, and there is no water and there is just vodka, of course, it's going to have a big effect. It's going to maybe make someone feel a bit drunk. But if I stick it in 5 liters of water, that one shot of vodka, that's what's my competitive inhibitor, it's not going to have an effect, or like such a minimal effect that it's not stopping me hit my Vmax anymore. So when we think about competitive inhibition, we can still reach the maximum amount, and they can recover by increasing the amount of substrate. So that's where it gets interesting with enzymes. And enzymes, like we're going to speak more about them when we talk about respiration and the types of enzymes involved in those reactions. But to put it all together, we want to think about like some keywords. An enzyme acts for its active site. It gets destroyed by denaturation, which is pH and temperature. They bind by induced fit or lock and key, and active site is blocked by inhibitors, and it can be a competitive inhibitor, like competing for the active site. Non-competitive would be going for an allosteric site, which obviously not in this diagram here, but an enzyme works by lowering our activation energy. So as with all the other interactive lessons, I've got a few activities for you. Here's the answers, but you can drag and drop them yourself. And some multiple choice questions to finish off. And there's 10 here. And there's also another multiple choice test on my website that is free for you to access with instant feedback. Let's just do the first two. So how does an enzyme increase the rate of reaction? You can pause it, think about it, or by lowering the activation energy. And what's neat about these is you get little feedback as you go. And what does the induced fit model state? Both the enzyme, I don't. And how does induced fit binding help catalysis? B again. It strains in the double bonds. Okay, so feel free to finish these on your own. And remember to check out my other site, Cramly, which is down at the bottom, if you want some help with some flashcard revision for your tests coming up. And of course, please check out all the free resources on my site. I hope they're useful, and feel free to make a comment about what you would like to see appear next. Okay, thanks for listening if you got this far, and ciao for now.